Hello everyone and welcome to Pleistocene Park. My name is Jacob Kostecke and I'll be your guide on this research journey. I'll be showcasing the research I've been doing for the past year. A special thanks to my research professor, Dr. Wei-Long Hao. Before we begin, I want to set the stage. What was the Pleistocene era like? It spanned from about 2.5 million to 10,000 years ago. The Earth was frozen over and experienced many great ice ages, the last just being 10,000 years ago. During this period, Earth saw an explosion of biodiversity, ranging from woolly mammoths to giant sloths and apex predators such as saber-tooth tigers and giant cave bears. There is a current knowledge gap in the field. The current research is insubstantial. What caused the Pleistocene extinction? There is a dispute in the scientific community about what led to the extinction. Some theories point to humans overhunting, the environment getting warmer, creating a loss of habitat, and even a giant asteroid slamming into Earth. However, I want to look at genetic mutations and the possible role they may have played. It is known that mutations can lead to extinction, and they offer a unique, under-researched perspective. This brings us to the hypothesis. I hypothesize that accelerated genetic mutation plays a major role in the extinction of mega mammals during the Pleistocene era. Through our hypothesis comes two aims, the first being aim 1, in which I determine whether extinct mammals have underwent accelerated evolution compared to their closest relatives. If the extinct mammals underwent accelerated evolution, then all genomic regions would have high substitution rates. For our second aim comes aim 2, in which I determine whether all extinct species suffered a prolonged small population size. This is achieved by comparing synonymous versus non-synonymous substitutions between extinct and living species. A ratio is then calculated by having non-synonymous substitutions over synonymous substitutions. Next, the methods of my research. I decided to use the powerhouse of the cell, the mitochondria, specifically its genome. They are an excellent vessel to do my research because mitochondrial genomes are abundant in preserved specimens. They are more well preserved and in smaller size than the larger, fragmented nuclear genomes. And lastly, they are perfect in viewing traceable mutation patterns that can easily be interpreted. Using software, Geneious Prime, and Python, I looked at 25 extinct species with 13 coding genes each. I used BLAST to find the 3 closest living relatives, constructed phylogenetic trees to see their relationship, and I made alignments of the extinct species with the first and third living relatives. The results, dn over ds ratio. The higher dn over ds ratio is consistent with the more relaxed selection due to prolonged small population size. The data on the right shows extinct species had a higher dn over ds ratio of mutations than the living. Extinct also had more non-synonymous mutations. We can conclude that extinct species underwent accelerated evolution under a prolonged small population before dying out.